welcome to this explainer. Today, we're looking at something absolutely extraordinary. When we think of insulin today, we rightly think of it as a life-saving daily medication for millions of people. But, it is actually so much more than that. Insulin is literally the foundational blueprint that single-handedly birthed the modern, multi-billion-dollar biopharmaceutical industry. The story of insulin is the textbook case of how medical science evolved from crude, almost unimaginable early experiments into the highly rational, hyper-advanced biotech manufacturing that completely defines our world today. Okay, let's dive into this by looking at where it all started, because the contrast here is just wild. Back in 1889, researchers von Mering and Minkowski discovered that if they removed a dog's pancreas, the dog immediately became diabetic. Now, that sparked a massive global hunt for the internal secretion of the pancreas. But it is a very long way from an 1889 lab tying off animal organs to the towering steel bioreactors of today's industrial scale. This entire explainer is really about that escalating journey of relentless problem-solving and innovation.
So let's kick things off with Section 1, the crude extract era, running from 1889 to 1922, where the major hurdles were extraction and scale.
For 30 long years after that initial 1889 discovery, scientists globally knew the pancreas held the key to treating diabetes, but nearly everyone who tried to extract the active ingredient totally failed. Why? Well, because the pancreas is a digestive organ. It's absolutely packed with pancreatic proteases. These are enzymes designed to break things down, and they simply destroyed the active principle before it could even be extracted. It was like trying to disarm a bomb with a built-in self-destruct button. That brings us to the summer of 1921 in Toronto. Frederick Banting and a medical student named Charles Best, working in J.J.R. MacLeod's lab, tried a brand new approach. They surgically ligated or tied off the pancreatic ducts in dogs to intentionally atrophy the digestive exocrine tissue. Only then did they extract the remaining islets. And it worked. They managed to keep a depancreatized dog alive for 70 days. But the harsh pharma reality was that this extract was incredibly dirty. We're talking a messy soup of proteins, lipids, and leftover pancreatic enzymes. And that messiness had severe real-world consequences. On January 11, 1922, a 14-year-old boy named Leonard Thompson received the very first human dose of this extract. The result? A modest, incredibly promising drop in his blood glucose. But it also caused a sterile abscess at the injection site and a highly dangerous fever. This really highlights the massive physical stakes of using an active pharmaceutical ingredient with absolutely zero downstream purification. They essentially had a miracle on their hands, but it was just far too toxic to use regularly. The biochemical breakthrough came just days later. A biochemist named James Collip was brought in, and he introduced an acid-alcohol extraction method. The acid inactivated those destructive proteases we talked about, and the alcohol precipitated out the contaminating proteins. By January 23rd, Leonard Thompson got Collip's cleaner extract, and it worked dramatically with no major toxicity. But a university lab couldn't scale this for the entire world. Eli Lilly stepped in to industrialize it, and their chemists introduced a massive game changer: isoelectric precipitation. By precisely adjusting the pH to about 5.4, which is the exact point where insulin becomes insoluble, it drops right out of the solution perfectly. This single move spiked yields by 10 to 100-fold, and it's a foundational processing step still used in pharma today. Moving right along to Section 2, perfecting formulation and purity, where we'll look at the timing and purity problems from 1926 up to the 1970s. Now, what's really interesting about this slide is that even once they had a cleaner, usable drug, early regular soluble insulin was a complete pharmacokinetic nightmare. It had massive timing issues, it kicked in after 30 minutes, peaked in just 1 to 2 hours, and was entirely gone from the body in 6 to 8 hours. That meant a diabetic patient required 3 to 4 painful injections every single day just to survive. Solving this timing problem brings Denmark right to the center of our story. In 1936, Hans Christian Hagedorn mixed insulin with, of all completely unexpected things, protamine derived from fish sperm. I know, bizarre, right? But it formed a complex that dissolved much slower in the body. By 1946, Crayenbull found the precise isophane point, the exact pH where protamine and insulin co-precipitate without excess, creating NPH, or neutral protamine Hagedorn. It was the world's very first rationally engineered slow-release depot. And in the 1950s, the other Danish powerhouse, Novo, developed the Lente series using zinc suspensions to slow the release even further. But timing wasn't the only issue. Purity was still a huge hurdle. See, for 40 years, the world relied on animal insulin from pigs and cows. Pig insulin differs from human insulin by just one amino acid, and cow insulin differs by 3. That sounds tiny, but along with impurities like proinsulin and high molecular weight proteins, it was enough to cause severe immune reactions, lipodystrophy, and insulin resistance, where patients literally developed anti-insulin antibodies. The holy grail here was theoretically perfect, completely biocompatible human insulin. Getting closer to that grail required parallel leaps in both science and manufacturing. Analytically, Frederick Sanger fully sequenced insulin in 1951, which was the first protein ever sequenced, by the way, and Dorothy Hodgkin solved its 3D crystal structure in 1969. On the pharma side, Abel had already introduced zinc crystallization as a purification step back in 1926, but by the 1970s, Novo Nordisk took this to a whole new level. They used those analytical blueprints to introduce monocomponent insulin. By utilizing gel and ion exchange chromatography, they dropped impurities down to less than 10 parts per million. Immunogenicity dropped dramatically, setting the gold standard for how polypeptide drugs are purified to this very day. Still, they desperately needed the exact human sequence. Before modern synthetic biology even existed, enzymologists built a brilliant semi-synthetic bridge. In 1978, they used an enzyme called trypsin to literally swap out that one differing amino acid in pig insulin, replacing it with the human amino acid. This successfully proved that human insulin was indeed far less immunogenic. But there was a glaring, fatal flaw to this bridge. You still relied on millions of animal organs to get your starting material. Which brings us to Section 3, the bottleneck, and the very real crisis of scaling the supply. The tone of the history shifts right here, from scientific curiosity to a looming absolute crisis. Relying on animals for a life-saving drug was fundamentally, mathematically unsustainable for a rapidly growing diabetic population around the world. So the crucial point is this staggering number right here, 56 million. By the 1970s, Eli Lilly alone required 56 million pig and cattle pancreases every single year just to supply the United States market with insulin. The supply chain was incredibly fragile. It was biologically and logistically maxed out. They didn't just need an upgrade, they needed a total miracle that didn't rely on slaughterhouses. Enter Section 4, the recombinant revolution. This was the ultimate solution that played out between 1976 and 1982. This revolution is unequivocally the most important pivot in pharmaceutical history. The context is vital here, because up until 1977, nobody on Earth even knew for sure if chemically synthesized human genes could actually function inside living bacteria. It was purely theoretical. Yet the pace of this timeline was insanely fast. In April 1976, Herbert Boyer and Robert Swanson founded Genentech. By 1977, they successfully synthesized somatostatin, the first synthetic human protein ever made in bacteria. And less than 6 years after the company was founded, in October 1982, the FDA officially approved Humulin. It was the very first recombinant pharmaceutical approved in the United States. And this brilliantly illustrates how they actually pulled it off. The mRNA sequence for insulin was entirely unknown, so Genentech scientists had to back-translate it from the protein sequence. Insulin consists of 2 chains, A and B. So they hacked E. coli by creating 2 separate bacterial strains. Step 1. One strain manufactured the A chain, and the other manufactured the B chain, both completely disguised as a fusion protein so the bacteria wouldn't destroy them. Step 2. They cleaved and purified these chains separately. Step 3. They joined them together using air oxidation to form the exact disulfide bonds found in human insulin. Absolutely brilliant. Now, Genentech proved it could be done, but early yields were pretty low, so they licensed the tech to Eli Lilly, who threw their massive weight into scaling the revolution. Lilly industrialized the bacterial fermentation, they mastered the incredibly complex recovery and refolding processes, and built large-scale chromatography plants. Interestingly, later processes even evolved from using E. coli to using yeast, specifically Saccharomyces cerevisiae, because it turns out yeast is naturally much more efficient at folding proinsulin. Finally, we reach Section 5, the birth of modern biopharma, and its enduring legacy. Let's move to this and see how this builds the ultimate pharma innovation template. Looking back at this entire 100-year historical arc, we can clearly see how this specific, relentless journey to solve the insulin problem laid the unshakable foundation for absolutely all biologic drugs on the market today. The insulin journey proved 3 critical things. 1. Supply security. We moved from an animal organ bottleneck to infinite, reproducible fermenters. 2. Biocompatibility.
By making the true human sequence, we eliminated dangerous immune reactions forever. And 3. Platform creation. They proved that bacteria and yeast could be reliable, GMP-certified factories for therapeutics. This exact template was copied verbatim for human growth hormone in 1982, and then for interferons, monoclonal antibodies, and even the incredibly popular modern GLP-1 analogs we see today. It all traces back to this model.
And this perfectly explains the modern landscape. The reason legacy companies like Novo Nordisk and Eli Lilly still entirely dominate the global diabetes market is actually really simple. They quite literally built the underlying mechanics of the industry. They own 100 years of proprietary process knowledge, from the earliest isoelectric purification to zinc hexamer stabilization to mastering crystallization all the way up to today's recombinant analog engineering. They didn't just make the drug, they completely invented the way the drug is made.
We started with scientists tying off a dog's pancreas in the late 1800s, and we ended up creating the entire biopharmaceutical industry. Which leaves us with this provocative final thought. If hacking E. coli to make insulin built the last century of medicine, what biological limits will we engineer away in the next 100 years? The blueprint has been drawn, but the incredible story of biopharma is really just beginning. Thank you so much for joining me for this explainer, and I hope it changes the way you look at the medicine in our world today.